So far in this course, we've been focusing on the innate arm of the immune system. But there is a second component to the human immune system, which is the adaptive response. And this is what we will be covering over the next few videos. Now remember, although we may teach these systems individually, they are constantly working together to help mount the most effective immune response possible, keeping us fit and healthy. In this introductory video on the adaptive response, we're going to take a broad overview of the key components we will be covering in the remainder of this course. If we compare and contrast the innate and adaptive responses, we can see some clear differences straight away. The innate response is extremely rapid, sometimes responding within minutes. And as we discussed, it is relatively non-specific, driven by the pattern recognition molecules. So although it can tell the difference between, say, a bacterium and a virus, it's unable to tell the difference between the multiple different types of bacteria that we might encounter as part of our daily lives. So in that respect, the specificity is very limited. The cell surface proteins and soluble mediators of the innate response are germline encoded. So this means they have been inherited from your mother and father as they are expressed by them. And there is a wide range of cellular and humoral components, and these inflammatory mediators can be directly or indirectly toxic to the pathogens. The adaptive system is slower to respond, taking somewhere between 7 and 10 days to kick in on the first encounter to that pathogen. However, the advantage to this system is a high degree of specificity and targeting to the threat that is being encountered, and this is why we were able to develop an immune response to the novel coronavirus that causes COVID-19, despite the fact we've never encountered it before. Unlike the innate system, which is germline encoded, the adaptive system rearranges the genetic information that is inherited from your parents in a way that is highly tailored to you as an individual through a process of somatic rearrangement. So there are 3 key features of the adaptive immune response, which are diversity, clonality, and memory. And central to the adaptive response are the B and T lymphocytes. When we are talking about the adaptive immune response, we are mainly talking about those cells that are derived from the lymphoid lineage of hematopoiesis, so the B and T lymphocytes. But we also include dendritic cells as part of the adaptive response, although these really act as a link between the 2 arms of the immune system. Macrophages are part of the innate immune system, but also help link to the adaptive response, as like dendritic cells, they are able to process antigens and present them on their surface, so they belong to a class of cells called antigen-presenting cells. The adaptive response centres around 3 main cell populations: antigen-presenting cells, or APCs, which include the dendritic cells and macrophages we just described, and these can be identified by the fact that they present a molecule called Major Histocompatibility Complex, or MHC class II, on their surface. All nucleated cells in the body express MHC class I, so that is every cell except red blood cells and platelets, but only the professional antigen-presenting cells express class II MHC. And these MHC molecules are fundamental for initiating the adaptive immune response, and help bring the specificity associated with that response. Then we have our T lymphocytes, which are produced in the bone marrow but mature in the thymus, and they migrate to the lymph nodes once they've matured. We have 2 broad subclasses of T lymphocytes, which are based on the presence of a surface protein called CD4 and CD8. CD4 T lymphocytes are also called helper T cells, while CD8 T lymphocytes are also known as cytotoxic T cells. We will learn more about their different roles and some of the subsets within these 2 families as we move through the course. T lymphocytes have a unique antigen recognition receptor on their surface called the T cell receptor, or TCR, and only T cells express this receptor naturally. T cells recognize the antigen that they are specific for, or their cognate antigen, via this TCR, only when it is expressed via MHC. The T cells are unable to recognize free antigens. They have to be associated with the MHC class I or class II molecules on these antigen-presenting cells. In contrast, B lymphocytes are produced and mature in the bone marrow in humans, and are able to recognize free antigen, so they do not need to be bound to the MHC molecule on an antigen-presenting cell. And in fact, B cells are themselves professional antigen-presenting cells, expressing MHC class II, so they are able to bind and subsequently present the antigen they recognize. They do this via the B cell receptor, or BCR, which can be membrane-bound, or if it occurs as a soluble protein, we know this as antibody. And again, only B lymphocytes can express the BCR and produce antibodies. An important feature when we consider the adaptive immune system is immunological memory, and this concept of immune memory is entirely dependent on these T and B lymphocytes. As we go through the rest of this course, we will talk about naive and memory cells, so it is worth defining what we mean by these terms. We describe a B or a T lymphocyte as naive if it has not yet encountered its cognate antigen, or the antigen it specifically recognizes. Once a naive cell has made that initial encounter and has matured or become activated, we describe it potentially as a memory B or T cell, because it now has the capacity to respond much more quickly and robustly should it come across the same antigen again. So CD4 positive T cells or helper T cells will reproduce a variety of cytokines to help direct the immune response. CD8 positive or cytotoxic T cells will become activated against cells displaying a complementary antigen in their MHC molecule and initiate cell lysis, whilst B lymphocytes will differentiate into plasma cells and start producing vast quantities of antibody. Now, SARS-CoV-2 is a really nice example of this immune memory, as prior to December 2019, as far as we are aware, no one on Earth had previously encountered the novel coronavirus. But as the pandemic swept the globe, more and more people were infected by the virus for the first time and started to develop immune memory. Once the vaccination program started, we were able to induce that first exposure to generate memory in a greater number of people. Immune memory and vaccination are based on the principles of a primary and secondary immune response. So on first exposure to an antigen, be that an infection or the first dose of a vaccine, the adaptive immune response is quite weak and slow to respond. However, after that first exposure, some of the T and B lymphocytes that were specific to that antigen become long-lived memory cells. So if we encounter that same antigen again, either from a second dose of the vaccine, or if we are unfortunate enough to be reinfected by the same pathogen, the adaptive immune system kicks into gear much faster and produces a more robust response, so we clear that threat much more effectively, and we may not even realise that we had been infected. Just to note though, this enhanced secondary response is specific to that original threat only. So, if by chance at the same time as we were mounting a secondary response to pathogen A, we were infected by pathogen B, which we have never seen before, we would only make a primary response to that second pathogen. And this is the concept of specificity, which is a hallmark of the adaptive immune response, and this specificity is due to the memory T and B lymphocytes. When we introduce the lymphoid lineage, we oversimplify the diagram a bit by only defining B and T lymphocytes. But as we have just discussed, there are different types of cells within these groups. When a naive B lymphocyte is activated, it either becomes a plasma cell, which is responsible for producing antibodies, or it becomes a memory cell, which is long-lived and provides part of that immune memory we just discussed. T cells can be broadly divided into three main groups: CD4 positive helper T cells, CD8 positive cytotoxic T cells, and regulatory T cells, which play a role in controlling or regulating the immune response to ensure it is appropriate. As with B lymphocytes, the T cells are also capable of forming memory cells after being activated by their cognate peptide. And this population of memory T cells can respond far more effectively upon secondary exposure to a pathogen. Key to the activation of T cells is the dendritic cell, which acts as a link between the innate and adaptive immune systems. Dendritic cells are constantly sampling their environment, and if they detect a threat, they phagocytose it and present part of that threat, which we call an antigen, on their surface in the major histocompatibility complex. They then migrate to a lymph node and present that antigen to the T cells, allowing them to see that threat and become activated. And we will discuss this process in a lot more detail in a future video. You may have picked up on the fact that the adaptive response has both cellular and soluble components, just like the innate system. And while the adaptive arm produces its own cytokines and chemokines, the key soluble component of the adaptive response are the antibodies that are produced by these plasma cells. Now, antibodies have multiple functions. The first is neutralization of the threat. Antibodies are quite large proteins, so when they bind to antigens, they make them much larger than they would be normally, and this can prevent the pathogen or toxin from entering cells or causing damage. Secondly, opsonization. We've already mentioned this, but antibodies mark the antigen for removal. Thirdly, agglutination. So if multiple antibodies bind the same antigens, they can form large complexes that are very attractive to phagocytes. And this combines the neutralization and opsonization properties to help clear the threat more effectively. Fourthly, antibodies can help recruit innate immune cells to carry out their functions, as well as activating the complement system via the classical pathway. Now, you may have noticed that these first functions relate primarily to the innate immune response. So despite being produced as part of the adaptive system, they provide a link between the two arms of the immune system. The last function relates to the adaptive arm, however, and this is to aid T lymphocyte recruitment and activation. So you can see that antibodies have a wide range of functions to help protect us. Beyond this, plasma from patients who have recovered from a disease, for example Ebola or COVID-19, contains antibodies against that pathogen and can be given to new patients to provide short-term protection while their own immune system kicks in. This is only a short-term option as the antibodies, being proteins, are removed from the circulation over time. But this treatment option, which we call convalescent plasma therapy, can help save the lives of critically ill patients, although the effectiveness can vary between different diseases. If all else fails, we move on to the battle royale of the immune system. Imagine a scenario. Our physical barriers have been breached. The innate immune system tried to repel the attackers, but they were overwhelmed by the sheer number of threats. The B lymphocytes produced antibodies to try and keep everything in check, but the pathogen still managed to invade our cells. So it's time to wheel out the ultimate weapon of our immune system, the T lymphocytes. Just like Vlad the Impaler used to decorate himself with the bones of his victims, many cells of the body do something similar, decorating themselves with either parts of the pathogens they have engulfed or the thing that is infecting them. So, for example, phagocytes like macrophages take up a pathogen and then present part of that pathogen on their surface. Now, all nucleated cells in our bodies can present antigens in one way or another, as they all express MHC class I. But we also have these professional antigen-presenting cells, our macrophages, our dendritic cells, and our B lymphocytes, which express MHC class II and play a central role in the immune response. Healthy cells naturally express endogenous or self-peptides on their surface, an MHC class I, that tells any circulating T lymphocyte they might come into contact with that everything is okay and that they are fit and healthy. If, however, a cell becomes infected or perhaps cancerous, then that cell will present either part of the invading pathogen or a modified self-peptide, which alerts the circulating T cells that there is a problem. Essentially, the cell is showing the T cell what is going on inside it, and is asking to be killed for the benefit of the body. The professional antigen-presenting cells are our macrophages, dendritic cells, and B lymphocytes, and all these cells are able to take up pathogens and then express fragments of those pathogens in the groove of their major histocompatibility complex class II molecules. So instead of endogenous peptides, MHC class II presents exogenous or non-self peptides. These antigen-presenting cells, or APCs, play a fundamental role in adaptive immunity by activating CD4 helper T lymphocytes, which in turn produce cytokines that activate other cells, and by driving the maturation of CD8 T lymphocytes, induce cytotoxic killing. The major histocompatibility complex, or MHC, plays a key role in allowing the immune system to identify self from non-self and pathogens that can induce harm. So they're central to this stranger danger model of immunity. The MHC is encoded by a large cluster of genes on the short arm of chromosome 6. It contains millions of base pairs, producing over 200 genes and sweeter genes, and it is the most polymorphic gene cluster in the human genome. The genes encode the human leukocyte antigens, or HLAs, which are expressed on the surface of our cells and make up our tissue type. Structurally, the MHCs are similar, but they have some important differences. The MHC class 1 is made up of 3 alpha domains in a chain, a transmembrane domain, and a cytoplasmic anchor. A beta-2 microglobulin domain associates with the alpha domains but is not covalently bound. The peptide binding groove is made up of the alpha-1 and alpha-2 domains and can bind an 8 to 10 amino acid peptide. The MHC class 2 is made up of 2 non-covalently bound chains called the alpha chain or the beta chain. The peptide binding groove formed between the alpha-1 and the beta-1 domains can present a 13 to 18 amino acid long peptide, so it's slightly longer for MHC class 2. So that is a brief introduction to the main features of the adaptive immune system. Of course, the precise processes that underpin this activation are a lot more complicated, and over the course of the next few videos, we will take a closer look at the roles of dendritic cells, T and B lymphocytes, to build up a much better picture of adaptive immunity.